Welcome to Dr. CM Edcast, your go-to resource for medical learning. Through discussions, case studies, tutorials, and debates, we help you learn, grow, serve.
Okay, so here's our game plan. We'll kick things off with the genetic switch that gets this whole cascade started. Then we'll dive into how the initial indifferent gonads are formed. After that, we'll compare the development of the testis and the ovary, see how the internal plumbing, the genital ducts, follows their lead, and we'll wrap things up by looking at the descent of the gonads and the formation of the external genitalia.
All right, first things first. We have to understand the genetic determinants of sex. This is really the single instruction that sets one of two incredibly complex developmental pathways into motion. Now, you might think sex differentiation is this hugely complicated process, and it is, but it all really boils down to one master switch, the SRY gene. It's located right there on the Y chromosome. The protein it makes acts as what we call a transcription factor. Basically, it tells other genes when to turn on or off. And in this case, it's the testis determining factor, and it's what kicks off the entire chain reaction leading to male development. So here's the bottom line. If a Y chromosome is present, its SRY gene is going to direct that early indifferent gonad to become a testis. But if that influence is absent, say in an XX individual, that very same indifferent structure will naturally develop into an ovary. It's a fundamental fork in the road. Okay, with that genetic switch in mind, let's rewind a bit. Let's see how the actual raw materials for the reproductive system get established in the early embryo, long before that SRY gene even has a chance to act. This timeline is great because it lays out the key milestones for us. You can see that genetic sex is decided right at fertilization, but for weeks the actual structures remain indifferent or neutral. The real action starts around week 3 when the primordial germ cells show up, and it all comes to a head in the 7th week when we finally see visible differentiation. So where does this all begin? It starts right here. In a tiny 3-week-old embryo, the primordial germ cells. These are the actual precursors that will one day become sperm or eggs, first appear in the wall of the yolk sac, a structure outside the main embryo. Now, from that yolk sac, these cells go on a pretty incredible journey. They actually migrate along the wall of the hindgut and travel through the dorsal mesentery to reach their final destination, the genital ridges. Think of these ridges as thickenings of tissue on the back wall of the developing abdomen. So once they arrive around the 6th week, these germ cells signal the tissue of the genital ridge to grow and form these things called primitive sex cords. This combination, the migrating germ cells and the local tissue, creates what we call the indifferent gonad. It's the blueprint, a structure that holds the potential to become either a testis or an ovary. And this brings us to a really pivotal moment in our story, the Great Divergence. This is the point where that indifferent gonad, following its genetic instructions, commits to one of two very, very different developmental paths. This slide gives us a fantastic preview of those two potential outcomes. On the left, you can see the path towards a testis, where all the action is happening deep inside the core, or medulla, of the gonads. And on the right, you see the path to an ovary, where development is focused on the outer layer, the cortex. Let's break down each of these paths right now. So, under the influence of that SRY gene, the primitive sex cords keep growing and push deep into the medulla, the very core of the gonad. They form the testis cords and a network called the rete testis. Then, a thick fibrous capsule, the tunica albuginea, forms, walling these cords off from the surface. And here's the crucial part. By the 8th week, Leydig cells show up and start pumping out testosterone, the hormone that's going to direct the rest of male development. Now, what happens in an XX embryo without that SRY signal? Well, it's a completely different story. Those initial deep medullary cords just degenerate. Instead, the surface tissue grows and forms a second generation of cords called cortical cords. These stay close to the surface, and by the 4th month, they split up into little clusters that surround each primordial germ cell, forming the primordial follicles. You know, what I love about this slide is how it just boils everything down for us. In an XY embryo, development is medullary. It happens in the core. The cortex regresses. In an XX embryo, it's the total opposite. The medulla regresses, and the important functional part of the ovary develops from those cortical cords right near the surface. Okay, so the gonads have picked their path, And in the case of the testis, they're already making hormones. So now, they're ready to start directing the next phase, the development of the internal plumbing, the genital ducts. What's pretty wild is that at first, every single embryo is equipped with 2 complete sets of ducts running side by side. You've got the mesonephric or Wolffian ducts and the paramesonephric or Mullerian ducts. So the potential for either male or female internal tracts is there in everyone. And this is where those hormones really come into play. In males, the testes release 2 key things. First, testosterone tells the Wolffian duct, hey, you're up, and it develops into structures like the epididymis and ductus deferens. Second, they release anti-Mullerian hormone, which actively causes the Mullerian duct to degenerate. Now, in females, there's no testosterone and no AMH, so the Wolffian duct just degenerates on its own, and the Mullerian duct is free to develop into the uterine tubes, the uterus, and the upper part of the vagina. All right, we're in the home stretch now. Let's look at the final positioning of the gonads and the formation of the external structures. And just like everything else we've seen, these also start from a common, indifferent state. So the gonads can't stay where they started, high up on the back wall of the abdomen. For males, this is a long journey. The testes have to descend down through the inguinal canal to reach the scrotum, and this usually happens by birth. For females, the descent is much, much shorter. The ovaries just settle right below the rim of the true pelvis. Without that strong influence of testosterone, and with estrogens now leading the way, the external structures develop along the female pathway. The genital tubercle becomes the clitoris, the urethral folds stay unfused and become the labia minora, and the genital swellings get bigger to form the labia majora. And here's a great visual of that process. On the left, at 5 months, you can still make out the individual parts, the tubercle, the folds, and the swellings. But by the time of birth, on the right, they've matured into their final recognizable forms. So we come back to this central question: how does one single blueprint create 2 totally distinct systems? Well, the answer is just this beautiful cascade of events. You have cell migration, precise hormonal signals, and targeted gene expression, all triggered by that one single genetic switch. It really is a remarkable testament to the efficiency and honestly the elegance of developmental biology.